Welcome back to the last topic in this series on data integrity, which aims to provide practical tips towards data integrity. In the last two topics, we talked about ALCOAplus and FAIR in a more theoretical manner. In this topic, we will talk about the procedures you could establish in your lab work to apply them. This slide gives an overview of some of the different aspects we can apply to increase data integrity. We will talk about how to establish a unique study identifier, templates and why it's beneficial to use them as often as possible. Establishing routines can help to reduce errors and achieve more in less time. The data management plan is a very useful document, especially for collaborations, to clarify best documentation practices. Alignment between all lab members also helps productivity and ensures data integrity. And finally, we will briefly talk about how to make protocols reproducible. Establishing a unique study or experimental ideas is a very useful asset for any lab. So what is it? It's an identifier for each experiment or study to unambiguously identify it. The benefits certainly outweigh the hassle of implementing it, because you can find experiments much faster, you can cross-link them between different files or documents, and it ultimately creates transparency in the documentation system, something that is highly desirable. How do we implement such identifiers? For example, unique IDs can be defined by consecutive numbers, or, if in place, an automated system that creates identifiers automatically. I would like to present three different approaches, shown here, which shall only provide ideas for thoughts. For a general approach, one could start with a date, followed by the first letter of your first name, together with your last name separated by a hyphen. This is followed by a short method description and some free text providing information about the experiment. The example shows the date, 10th of September 2021, the first letters of the name Björn Gerlach. Then Western plots as a short description of the method, and then a short description of the experiment, that HeLa cells were treated with bortezomib at this concentration range. In another example, we start with the initials of the researcher. Thereby, the file is directly attributed to the author. Now, an arbitrary numbering system is chosen to code the type of experiment. This is followed by a consecutive numbering system for each experiment carried out by this researcher. At the end of the ID system, there is again a short description of the experiment. For example, we start with the initials BGER, followed by 01, which is the code for Western blotting, followed by 05, indicating that this is the fifth experiment of this type carried out by this researcher. A short description of the experiment provides further identification. If you are using an automated system, like an electronic lab notebook, then this system might already provide an automatic identifier when designing the experiment or the study. And this can of course also be used to link the experiment throughout the lab. Just a brief note that the first two examples, since both of them have the initials of the author, these experiments are attributable and fulfill the first item of ALCOAplus, which is attributable. However, these are just ideas that have proven to work in practice. But in the end, you can create your own solution, and it must work for you. and even better if it works for the entire lab. Let's come to the next topic. Using templates routinely is a very powerful approach to support data integrity because it is often the case that different formats are used for protocols within the lab, and that makes it difficult for all users to understand them. Additionally, if documents need to be created from scratch, some important aspects are often forgotten, such as metadata. Therefore, the benefits of using templates increases consistency of documentation for experiments and studies. But it also reminds us about important steps that should not be forgotten. When creating templates, it is advisable to check out different resources as existing templates might be available. For example, ALCOAplus provides comprehensive templates for study protocols, methods and assays. They are linked below in the reference as examples. When creating templates or modifying existing ones, it is important to include as much detail as possible, so you end up with a comprehensive template. From experience, it is always easier to delete items later if you don't think you will use them, rather than remembering them and adding it to the document. Once the templates are created, it's important to ensure that all lab members understand the templates and know where to find them. For that, maybe a short training is necessary, and a common database can also be very helpful. It is also important to update the templates periodically. This can be systematic, like once per year or as needed. Establishing routines can also be very helpful because it might often be the case that a similar procedure is started from scratch each time. And this always requires time to think of how to do the procedure or what to name it. If routines are established, this will ultimately reduce errors and also free up time as you don't have to think about the next step. And with that, it's also less likely that you will forget things. Establishing routines is possible for different working steps, and I want to present just two brief examples. When starting a new document or file, or when setting up cell culture plates or storage boxes, always include a unique experimental ID or date or some identifier. The items will always be identifiable, thus ensuring that it is not forgotten. Another example, when performing a simple experiment, establish and follow a routine. Plan and write down or print the protocol in the office. Always have the printout with you when performing the experiment. This provides the ideal space to document any changes during the experiment, such as different volumes or samples, or when an incident happens. Once the experimental part is finished, this document can be taken back to the office and used for updating the lab notebook. Such a routine can ensure that no data is lost. A data management plan can be a useful tool, especially for research collaborations, and is now actually required by several funding organizations. Why are they needed? It often happens that management of data is unclear, especially in collaborative projects when data are generated at different sites, leading to confusion about who is responsible for maintaining and archiving the data. Another example concerns big data sets in projects. If large data sets are not properly organized, this will ultimately lead to loss of data. Having a clear defined and documented plan before the start of the project on management of the research helps save time and resources. How the data will be organized, agreeing on important aspects such as who is responsible for what, IP rights, the archiving platform. A data management plan also helps to describe how to achieve transparency of the data while the project is running. The data management plan describes the research data which will be acquired, how to work with it, and how to ensure long-term storage and findability. For setting up a data management plan, several tools are available to support the creation. The Data Steward Wizard is such a tool which requests to answer a comprehensive set of questions and even provides feedback. A little bit easier is the DMP online tool, provided by the second link here. Here you can find templates tailored for several funders, making it also easy to start. The data management plan should be considered as a living document, meaning that it is also needs to be updated and adjusted to new developments. Aligning between lab members and being in close communication can help to increase data integrity as well. It often happens that there is no common structure established in the lab, and everybody is working with their own procedures. Aligning on different levels will save time, especially when searching for data and experimental information. This can also avoid that information are lost when a person is leaving the lab. However, it also helps to avoid misunderstandings when all lab members agree on the same lab lingual. So what can lab members align on? They can agree on general data integrity principles and how to handle them in the lab. For example, how to store raw data and how to archive the entire experiment. Basically, on all aspects that we have discussed when talking about ALCOAplus. It could be agreed to use the same study ID format, which makes it easier to find and link experiments. We have talked about it before and how to set it up. We have also talked about using templates, which could be shared and used by all lab members. This could also be expanded to labeling the different resources in the lab in the same way, which will also provide additional clarity. This starts already when thinking of the date. Many people write the date differently, and having the same date throughout the lab will reduce confusion. To achieve such alignment, it is very important to establish a training for new lab members, because only if they have direct guidance from day one and integrate it in their work procedures, such an approach makes sense. Maintaining and improving such alignment can be supported by dedicating a few minutes of regular meeting to this topic. Such alignment can also be supported by a lab-wide infrastructure. For example, a joint database or a lab wiki may provide helpful guidance and a joint resource. If these things are not discussed in your lab, maybe you want to suggest it. This publication by Monja Baker describes 5 key aspects to write reproducible protocols and summarizes also some of the aspects that we talked about already. Document as you go, meaning do not wait until you want to publish your data or write your thesis to document your experiments, but think from the beginning about what is needed and document along your work. Write less, show more. This can indeed mean make a video of important steps of the experiment that might otherwise be unclear. For example, what means shaking? However, it can also mean to include a timeline in the protocol. Link, template, standardize. These are important points we have already extensively talked about. Create study IDs to link experiments. Use templates whenever possible and align in the lab to standardize as much as possible. Deposit and document reagents. Having full knowledge about the reagents and sharing them with the research community strongly supports reproducible research. And finally, communicate. That is a very important point, since communicating not only the scientific achievements, but also more details about the protocol among the lab members and the research community will help to increase transparency and data integrity. So this is a summary of these 6 steps we have talked about. And taking these suggestions by heart and implementing them in the lab, one way or another, will be a big step forward to achieve data integrity and have a sound system in place.